Dear viewers, today I will teach you gram-positive and gram-negative bacteria. So why these bacteria are called gram-positive and gram-negative bacteria? What is difference between these two bacteria we will cover today in this video. Gram is the name of scientist. He discovered a stain and that stain is crystal violet in color. Okay. So bacteria who attain this crystal violet color, they are called gram positive bacteria. You can see here the bacteria is in crystal violet color, while the bacteria who do not attain crystal violet color, they are called gram negative bacteria. You can see here the bacteria are pink color or red color. So what is the difference between this gram negative and gram positive bacteria? In gram-negative bacteria, you can see there is outer membrane present, and this outer membrane is absent in gram-positive bacteria. This outer membrane is made up of lipopolysaccharide, and in this lipopolysaccharide there is a channel present which is called porine, and in this outer membrane some embedded proteins are also present. Okay. So if you further zoom this outer membrane, you can see these thread-like structures are present. These thread-like structures are called O-specific side chain. So this whole outer membrane is absent in gram-positive bacteria. So this is the first difference. Second difference is this peptidoglycan chain. Only one chain is present in gram-negative bacteria, while you can see there are multiple layers of peptidoglycan chains is present in gram-positive bacteria. If you further zoom this chain, you can see this peptidoglycan chain is made up of teichoic acid and lipoteichoic acid. So this outer membrane and peptidoglycan chain create difference between gram-positive and gram-negative bacteria. So example of gram-positive bacteria is Staphylococcus and Streptococcus, while example of gram-negative bacteria is Escherichia coli and Salmonella typhi. Now we will study how gram staining works. So I already told you gram names come from the scientist name. His name was Hans Christian Gram, and he was Danish bacteriologist. In 1884, he developed this staining reagent. So how this gram staining works? So first you will take a glass slide. On this glass slide you will put some drop of bacterial culture. Now you will make a smear, and this smear you will make in circular form. Okay. After circular form of this smear you will do air drying. Okay. After air drying you will do flame drying. Now your smear will be fixed and your bacteria present is in dyed form. Okay. Now after fixation you will apply gram staining reagent on this slide. So first step is you will put crystal violet dye on it, and you keep it for 1 minute. After 1 minute you will rinse your slide with tap water or distilled water. The concept behind that is when you put crystal violet color in gram-positive bacteria, your peptidoglycan layer is exposed because there is no outer membrane to protect this. So this crystal violet is water soluble dye and it enter the exposed peptidoglycan layer. And in gram-negative bacteria there is a outer membrane present. So in that case your crystal violet dye do not enter, but some amount of dye still enter through porine channel present in gram-negative bacteria. Up to here it is complete. Second step is you add grams iodine, and you keep it again for 1 minute. Here you can see in this picture you are putting iodine on same slide and you keep it for 1 minute and you again wash it. So the concept behind this is iodine form complex with your crystal violet reagent. So now crystal violet and iodine complex is bigger in size. Now it is in bigger in size. It is not able to come out from peptidoglycan layer. Okay, so it will stay inside the cell. Now the third step is decolorizing the smear using 95% ethyl alcohol. So here we are using ethyl alcohol to wash excess of crystal violet present on slides. Okay, it is just for washing the excess of crystal violet color, and you have to wash it until your alcohol become colorless. Okay, so first use is decolorization. Second use of ethyl alcohol is dehydrating agent. So here you can see this is peptidoglycan layer. In first step you added crystal violet color. Okay, in second step you added iodine. Now crystal violet make complex with iodine, and this become very big in size and it is not able to come out. In third step you are adding ethyl alcohol. Ethyl alcohol, because of dehydrating reagent, it will shrink this peptidoglycan layer. Now this will get shrink, and this crystal violet and iodine complex is not able to come out and it will stay in this cell. Now in gram-negative bacteria, what happen in gram-negative bacteria? When you add ethyl alcohol, this outer membrane get destroyed. And similarly, if there is residual amount of crystal violet or iodine complex present here, this comes out because in presence of ethyl alcohol, this outer membrane as well as this peptidoglycan layer totally destroyed, and cell is not able to retain this crystal violet and iodine complex. Up to third point, everything is clear. Now the fourth point is you put safranin counter stain. Safranin has red color. Okay, now you will put safranin here. You already know this membrane is shrinked, so there is no space for entry of safranin into this peptidoglycan layer. So gram positive bacteria will not attain this red color. Okay, but here this membrane and this peptidoglycan is totally destroyed. So when you add safranin here, it will attain this red color or pink color. Okay, so this is the whole process of gram staining. So finally, you know the difference between gram positive and gram negative bacteria, why gram positive bacteria attain crystal violet color and why gram negative bacteria attain red or purple color. Thank you for watching the video.